So
hear
welcome
you.
everyone again to the Green Era Hub webinar series. My name is Barry Crosby and I'm one of the communication officers for the Green Era Hub project. If you've been here with us before, you'll know that the Green Era Hub project officially started in September 2022 as a coordination and support action of the European Commission under the Horizon Europe programme. And it currently represents 15 EU ERA-NETs, co-funds and self-sustained initiatives in the field of agri-food and biotechnology. We started these breakfast clubs here in October last year, and we've had four very successful series so far. The first was Circularity in Agriculture. The second series was related to biotechnology to advance sustainable solutions for bioeconomy. The third was Sustainable Food Systems, and the one that we just finished before Christmas was Precision Agriculture Technologies for a Sustainable Future. If you're interested in listening to the talks related to any of the topics from the series just mentioned, our recordings are now available to view on our YouTube channel. Now, for the next 3 months, we're focusing in on the topic of plant health and protection, where we'll be hosting a series of expert-led discussions aimed at fostering a deeper understanding of plant pathology, integrated pest management, and sustainable agricultural practices. And just a few housekeeping announcements. Please do feel free to type your question into the Q&A chat section, which I've enabled for everyone. I'm going to do my best to address as many questions as possible within the time that we have today, which is 1 hour. If at the end of the session you have questions for either of today's speakers, please do remember to address the speaker in the question. Now, today we have two panelists joining us, Dr. Rujing Wang and Dr. Khaled Jalua. And we will open our session this morning with Dr. Wang, a research scientist and currently head of the National Seed Herbarium at the Canadian Food Inspection Agency in Saskatchewan. In this role, Dr. Wang's work focuses on seed and grain quality assessment, and particularly the identification of crop and weed seed. A renowned expert in seed science, ecology, and botany, her research has significantly advanced our understanding of seed health and plant ecology, an important intervention which helps prevent plant health risks, including economic losses in crop production and the associated negative impacts on plant biodiversity and agricultural resources. Today, Dr. Wang's talk is entitled Development of a Seed Identification Guide and Database for Invasive Plants and Weedy Species. Dr. Wang, thank you very much for accepting our invitation for our webinar series. I'm going to stop sharing my screen now so you can start sharing your presentation. Thank you very much.
Everybody. It's my pleasure to get invited today to introduce the weed seed identification, which part of the plant protection in many countries' regulations. So CFI is a regulatory agency. We are science-based. However, we also enforce some of the Plant Protection Act for import and export. So our lab, our specialty is to seed testing and also grain quality analysis to make sure no invasive or regulated species to be spreaded through human trade activity. So today really is just concentrated on two issues. One is prevention. Another is how are we going to do this type of activity to detect and identify weed seed in human trade activity. So for weed seed introduction in the past, when we talk about pest prevention, usually in people's mind would be like disease, virus, or insect as well in people's mind. But actually, weed seed has been realized by many, many countries. It is also a pest for agricultural production. So Canadian Food Inspection, Canada did an estimation. This is many years ago. At least 2.2 billions each year lost by invasive plants for production. So the major weed spreading intentionally or unintentionally is by human trade activity. So weed seed contamination could through many processes. For example, seed for planting or seed as grain for consuming by human or by animal as a feed or a grain. It also could be pathway for weed seed spreading. So naturally, the natural spreading of weeds, they have their biological region or distance. However, human activity to facilitate this weed spreading is beyond their biological distance. So I gave some example. For example, the long distance spreading is by transportation. Then during the process, you could have some accident happens, then spreading the invasive seeds. And also the other concept is really plant spread by seeds. It's plants, when you see plants already established in a field, that means it's already spread it. So then just like we experienced before, like other pests or diseases. So weed seeds management, the most cost effective or most effective way or effective measure is to prevent the spread. Once it start established in the beginning, eradication could play a role, but later on, once they established, it's really difficult to get rid of them. So this is the reason the prevention, well known, is by regulation. So every country have a quarantine list. So those quarantine lists could have diseases, viruses, but now more and more country to add new species as weed species in their quarantine list. So in Canada, we also increased our quarantine list. The reason is the regulation to prevention is the most effective measure to stop spreading of invasive or noxious weeds. So in the past few years, like trade dispute, many case is around weed species because more and more country realize the weedy species could be a big threat for their agricultural production base. So here are some examples of the invasive species is more and more being realized by different production area. So these are examples for North America, Amaranthus palmeri, because it has high resistance to herbicides. So basically, when they established, it's hard to get rid of it. Even you use herbicide, they quickly developed herbicide resistance. So you couldn't get rid of them. So it has a big yield reduction in North America for or other regions for like a corn production, soybean. And this one is jointed gold grass. It's caused a big reduction of wheat production. So what I'm trying to say is weeds equals pest. So this idea, I hope this message is clear to everybody. Then in Canada, we have a weed seed order, which we have a newest regulation is in 2016. However, weed seed order in Canada, we started in 1920s. So this is very good. In Canada, we started this kind of prevention measures. So now we have 20 species added to plant regulated by Canada. So which is like a quarantine species for any commodity imported to Canada, you have to be free or practically free of regulated species indicated in Plant Protection Act. So similarly, other countries also increased their regulation for invasive species. So that posted very tough task for inspection or regulation enforcements is to identify seeds. So identify seeds is a very narrow niche of plant taxonomy, which is rarely being educated in our academia program or widely available knowledge for the society. I think plant identification, entomology, or other field subject is well known, but seed identification only known in seed testing laboratory. However, like I gave some example in UK or Canada, we have this testing technology being used in seed testing lab over 100 years, but still it's rarely been known. But now with the I shouldn't say globalized trade, it could be stopped sometime. But with trade, human activity, more country realize the weed seed identification technology. However, it's not widely available for many testing laboratories. So then we start to develop digital references. So there have many, but we started the digital references for collaboration. So we established ISMA website we call the International Seed Morphology Association. So that is a platform. Basically, anybody can submit a pest image or pest identification fact sheet or pest identification keys. So in this website, so for seed identification. So this digital tools is publicly accessible. It's a free access reference material. So we used a tool initially developed by Lucid, so which is Australia, developed a digital identification tool. It can be used a different kind of pest, but we used it to develop for weed seed identification. So anybody, because the time is limited here, anybody have interest, you can go there to this website to see all the resource availability to the public or testing laboratory. So the main product is a seed identification guide. However, there also have training resources for testing laboratory. So this is one of the product identification fact sheet. We listed different regulation and identification features and a gallery and tools people can use. Then we explored different kind of technology because seed identification very similar to entomology. We use majority identification method is morphology. So because morphology is quickly can be adopted by image analysis. So then we start the AI application. So use microscopic image, then we can do semi-auto identification to assist analysts, or we have the automatic sampling identification as well. So CFI developed a tool, there have a video demonstration. Basically, when you use digital identification, digital microscope, whatever the species, when you're doing the testing, you saw some seeds, you just take an image. Then after that, this we call it Nashe, this AI application, then it can cross and say what species it could be. So here tell you what species potentially it could be. One or two, they gave name and gave possibility. So this image tell you 93% possibility is a species one. And sometimes you can say this is 74 possibility is species three. So that's why we call it semi-auto. With that, then the analyst can further make verification what species it could be. The other system is a full automatic multi-spectrum imaging system, which take images, still images. However, the spectrum is more than visual lights, have a little bit of UV, have a little bit of infrared. So basically give the image more information of the images to computer, to train computer to know more information to get conclusion of their identification. So this is a visual image. Then you can see this is a multi-spectrum image. You can see the image type is very different, but both image can be used for um, identification. I don't know how much time I have. Please let me know. So you can see we use the two model to train identification. You can see the blue one is deep learning and this one is machine learning. So more and more computer model now is based on deep learning. Machine learning gradually phased out. So you can see the reason why. So we compared to model, definitely deep learning gave better result. So that's quick presentation and any questions maybe at the end, I'm not sure. You can go ahead and let me know.
Dr. Wang, thank you very much, I think, for such an interesting and insightful presentation. I really, really enjoyed it. I just want to remind everyone that the chat box and Q&A section is open now. So please do feel free, if you have a question, to type that into the chat box or Q&A section for Dr. Wang at any point. Dr. Wang, just while we're waiting for questions just to come in from our audience, I was just kind of fascinated when I was listening to your talk about the obvious kind of potential in terms of like education and outreach that such a project really kind of gives rise to. Um, and I was wondering about what strategies will be kind of employed, if any, to ensure that, like, for example, end users like farmers, landowners, maybe environmental managers are aware of this seed database and can kind of effectively use it.
Yeah, that's a good question because sorry I'm did I am still share the screen? I'm not sure. I should. Yeah.
Yeah, you
So
can stop sharing there if you don't mind, please.
yeah, I just want to say, um, so that's why I just want to show this website we
Oh,
developed.
sure.
Yeah. So I think that's we thought this could be a education process to let landowner to know, or even the producer, so they are not really know um, the weed seed is the root cause of the weeds. So we pay in a majority of the information, people talk about plants because when they see it, they realize it. So this prevention is easy to say and everybody understand it, but hard to really doing it. So this is my website is really try to do that to tell people. Um, so be aware. So seed is your main source of your weed management. So Like we have this fact sheet. How we did it is so you can have name or whatever. We also have a regulation. Like let people know. We put like because we are the lead, we mainly say, okay, so for Canada, this is prohibited noxious weeds. So if you click this one, you see all the prohibited species is there, then you click them and they will, so this would be the platform. This is public accessible for free. So you can see, okay, what it look like, where it's distributed, and what kind of general damage it can cause people. Then the identification, this part is more for private lab or seed testing lab. But before this is more like education. Like we tell what kind of regulation in place to prevent it and where it's distributed, and also the crop association, what kind of crop it potentially. So we hope this could be the way, but I think this shouldn't be the only way. I really promoting in university, like weed science, like even we associate with our neighbors is university. I talk to weed scientists. The more concentration is based on herbicides and develop a new herbicides. The reason for that is a funding. So all the research scientists or education sometimes go with where the funding come from, right? Herbicide by chemical company or whatever the reason. So they get more funding to do those kind of study where the supply chain or the trade, it should be obvious. However, the funding is limited for expertise of taxonomy. It doesn't matter for weed, for plants, for entomology, the taxonomy expertise is declining because people don't see the job, people don't see the funding. That's really the problem. So this should be the general education for, I don't know where to start. To increase the investment is a key to increase this prevention strategy to be known. Yeah, we only did this part to do the publication in a website to increase awareness. Yeah, I
That's
could.
excellent, Dr. Wang. Thank you so much for that. Just a comment and a question from an audience member. So it reads, great presentation. Are you aware of any research projects that have utilized the ISMA database? I've heard of people using iNaturalist for modeling population dynamics, and I'm wondering if there's similar potential to use these tools in research.
Definitely. I think the different kind of database, if we use AI, so first thing bottleneck is data. So there have Bugwood or US where I'm familiar. They have a load of plant images. Um, and plants, leaves, flowers. I think there have, if you go to the App Store, you definitely can find those apps. If you take a picture of flower, you can identify what plant species are. If you take a picture of plants or leaf, you can identify many software available. However, for seed, is big gap, is no data because seed is small. People, even I talk to plant taxonomists, they don't believe you can identify plant species by seeds. So that is a big gap there. So that's why seed image is rare. It's not sufficient to develop AI. So the awareness is number one to generate. So that's what ISMA try to do. We have a gallery. So we tell people if you see some seeds, but this gallery kind of thing, you must have like microscope. So it's not a general like a plant ID. You can get images from cell phone, the public what is called citizen science. So public can take images and send to Bugwood and whatever. Uh, then expert can fill it out to verify the authenticity to make sure they are valid data. However, for seed, unique problem. You see this one only 2 millimeter. So general citizen public, they couldn't take these images. But if institute, research institute, or university know about this, the data can provide it, then definitely it can increase the data, then the research program can going on. But I'm also aware, like in Netherlands, there have a seed atlas, and I think maybe other countries have a similar program as well, but very little. So definitely we need a more and more awareness of weed seeds. Weeds are spreaded by seeds. It should be such obvious, but it's not many people kind of aware of that. When they see plants, everybody pay attention and not seeds. Okay, I probably stop there, stop sharing as
Thanks
well.
very much, Dr. Wang. I think we just time for one last question. Just looking to the future, I'm just wondering, is there any potential for integrating the database with other databases on invasive species, such as those for maybe pests or pathogens, to create, I suppose, a more comprehensive management tool?
Yes, and I think we should have those kind of database to be shared. Um, um, I work with our CFI have a internal AI lab, so we are working on a structure to set up a database. So some private company also set up some database because they want to sell their machine or whatever. I keep telling our government or different organization, I think the data should be in a public hand, not in a private hand. Because if that is the case, then the problem of the future who can use that data. So it's really because if they develop a tool, use the public data, then they own that data, then the cost, I believe, going to be very high. So I'm talking my own company and government and different company, nonprofit, different seed testing association. I say those nonprofit organizations should set up this kind of database able to share. So ISMA is one of the nonprofit, but the problem is funding.
Mhm.
Yeah, so now is funding really is an issue, but I think we probably haven't tell the story big enough to let people realize. And also, only last 10 years, invasive plant species became a topic. Before, people didn't realize weeds are pests. They kind of know, but they didn't have a same level as, for example, disease
Yeah.
or something like some beetles. And public see that longhorn beetles, they feel that is a big threat. But when they see plants, they sometimes see even the gardener plant
Mhm.
those invasive species because they say, oh, they have beautiful flowers, right?
Yes. Yeah.
So this weed equals to pest. It's just recently
Mhm.
by regulation changes of different country, but I don't think publicly aware of this a big issue. The invasive species
Mhm.
take over plant species, take over habitat, take over increase the production cost, increase the herbicide applicants. So publicly hasn't have that clear understanding. I think the education like this is very important to deliver those message. When public aware of that, funding will come. Then this kind of research will be increased. The technology is ready. Limitation is funding.
Mhm. Dr. Wang, thank you so much for those wonderful insights. It's a really fascinating and in many ways a kind of a groundbreaking project. I wish you all the very best with it. It's fantastic just to hear all about it. Thank you very much also everybody for your questions, and of course thank you very much Dr. Wang for joining us today. It was a really excellent presentation. Now, joining us is Dr. Khalid Dejila. Just share my screen. There we go. So, Dr. Jalua is currently a senior researcher in plant protection at the Mediterranean Agronomic Institute of Bari in Italy, where he specializes in plant virology. Through his extensive research, Dr. Jalua has provided valuable insights into the mechanisms of viral infections, the molecular interactions between viruses and plants, and the pathways through which these diseases are spread. This research has not only deepened our knowledge of plant pathogen dynamics, but has also helped shape contemporary approaches to controlling and preventing viral outbreaks in agricultural systems. With over 100 published scientific papers, his work has become a cornerstone in the field, influencing both academic studies and practical applications in plant protection. His research has also contributed to the development of disease-resistant crop varieties, the enhancement of diagnostic tools, and the implementation of sustainable pest management practices. Dr. Jalua's talk today is entitled Detection and Identification of Severe Strains of Citrus Tristeza Virus. Dr. Jalua, thank you very much for accepting our invitation to our webinar series. We're absolutely delighted to have you with us today. So without further ado, the floor is yours. I'm going to stop sharing my screen now so you can start sharing yours. Thank you very much.
And you already introduced me, so let's go directly to the subject, which is related to the detection and identification of severe strains of citrus tristeza virus. This topic is now we are dealing with a project practically which we didn't end. So firstly, I would like to introduce us, most probably, most of you are not familiar with citrus. So just a quick introductions about the citrus disease. Citrus is most cultivated fruit crops in the world. It's widely distributed in the 5 continents. The problem is that citrus mainly their sanitary status is usually highly deteriorated by several pathogens, particularly bacteria and virus. And the citrus disease virus, which is one of the most destructive virus diseases over the world. This is when the sour orange is the rootstock. I will see why the sour orange, when the sour orange is a rootstock. So, relatively to the introductions of Citrus tristeza, it was mainly diffused through the propagating material. And here we have an example of the introductions of the Citrus tristeza virus in the Euro-Mediterranean area. In the past, the introductions of citrus was through seeds, and you can see here the trees were very big, and it was even difficult to harvest. But what happened around 1832? There was a first report of a fungi, of a Phytophthora, what we are calling Phytophthora disease. And it was a disaster in Portugal, in Madeira Islands, which were belonging to Portugal. And it was a disaster. What happened? It forced the farmers to use a sour orange, which is tolerant to rootstocks, which can be adaptable to citrus cultivated areas. So they succeeded to control the Phytophthora, but a new disease epidemic appeared. And the disease was related to a decline of the trees. It was thought first that it was an incompatibility of this rootstock. But in 1950, the citrus community adopted the word Tristeza based on most noticeable symptoms, which is the sad look of the trees. The main devastating field symptoms, as you can see here, are the quick decline of the trees and the seedling yellows, the seedling yellows and stem pitting. But we consider today the most severe forms are the stem pitting and the quick decline. The causal agents of this disease, as I said, is virus, is virus, which is a very big virus, is a flexible thread-like particles, and there is the size is 2000 nanometers per 11 nanometers. So it's accidentally long RNA, which predicts a high probability of sequence mutations. This virus is transmitted by different forms. First, by the man who is using if they are using, let's see, positive or diseased plant propagating materials. And insect vectors. This is the most important insect vectors, particularly the Toxoptera citricida, which is the brown citrus aphids, which can transmit the severe forms of this virus. What's the situation today? The situation today that we have a worldwide, as you can see here, distributions where citrus is cultivated. You can find the disease is a quarantine disease, and in order to control it, the use of plant material certifications as a preventive tool, and then the use of resistant or tolerant rootstocks. Here we have a list of these tolerant rootstocks because these tolerant rootstocks can control the disease. But this is true for the mild strains. In the last year, there was a lot of severe strains which appeared, and there was a breaking resistance for these rootstocks. So our problem is related now to these, let's see, severe strains. Severe strains which can be easily transmitted and quickly transmitted, but as I said, by the Toxoptera citricida, which is already present now in the Mediterranean area, in Madeira Islands, Canaries, and even in Portugal, in the mainland in Portugal. What's the situation today? The situation today that we have a lot of severe strains which were characterized, as you can see here, with the red color, and also others which can be considered as a mild strain. The problem is that in the last years, we have increasing a lot of genome sequences by using especially the high throughput sequencing and next generation sequencing. And we were facing a problem that the strain compositions is completely changed, and they are asking to include a new other strains of Tristeza. And there is also the fact that citrus species is grafted on different combinations of rootstocks. So we are in difficulties now. There is no clear correlations between genetic similarity and symptom severity. This is the situations in the Euro-Mediterranean area. As you can see here, most of them are the severe strains. Besides, of the severe strains were introduced beside the mild strains. So there is a need today. There is a need because even if they were introduced, but they were controlled and not to have a diffusions in the country. But today there is a need. There is a need of early detections of severe strains. Today we have it, but it's related to very lengthy protocols and using combinations of different techniques, serological, moleculars, and even still using biological indexing by observing appearance of symptoms on indicator plants. And you can see here, this is what I was telling. I was telling here that, sorry, I was telling here that we have, sorry, we have problems in order to detect early the severe strains or what they are here. And then it's very lengthy protocol. And you can see here, it's protocol including here in the case of Italy, different methods and one by eliminating by one, going from protocol to another in order to detect these severe strains. There was a need to make a project, and this project arrived as an fresco project under the umbrella of the EPO. This is the project where we are still working on it. It will be closed next year. And this is a rapid and efficient detection and identification of severe strains of citrus Tristeza virus. What's this, a fresco network? Fresco Network is an international network of organizations which is developing and managing phytosanitary research programs and use research provisions for policy developments and implementations, and maintain itself as a strong long-term network of research stakeholders. This is under the umbrella of, as I said, of the European Plant Protection Organizations. Our projects, the objective of projects was to simplify the diagnosis of monitoring of citrus strain by developing efficient diagnostic tools. 18 partners are participating to these projects, and this is what need to be done during the next year, or something was already done. We have the monitor and sampling plant material for the fields in order to identify the relevant collections on which we are working. This was already done, and then we have to do the design and analysis primers and props. It's this work is not concluded yet, where the Spanish colleagues are trying to evaluate these primers which were designed. And then we have the developments of protocol for the test performances studied and collections and analysis the results reported by the participants with the final reports. Concerning the CMBAR by institutions, we are coordinating these projects. And the main objectives for us is to improve the CTV monitoring scheme, especially in the field. And what we did in this framework, we did first a comparative analysis of extraction methods using different type of methods. We're using a semi-automated magnetic beads, which is a Maxwell robot from Promega. We also use a silica capture, as most of you know, for the detections of for the extraction of nucleic acids, and then the membrane printings. In this context, as you can see here, we set up different criteria. First, to analyze the RNA yields and purity, to analyze the quantitative PCR performance, and to make a survey in order to have an idea about the user friendliness and processing times of this, what we were testing, and also a survey on the cost, and obviously concluding by statistical analysis. we have what we did, we took also, so we have the collections of Tristeza strains in greenhouse in our institutions, and we took some of the silver strains and we started to analyze these aspects. Just to tell you that today the current CTV monitoring schemes is like this. We have the visual inspections in the fields, then we have the RNA extractions by different type of methods, RT-PCR, and then the real-time RT-PCR. Then we have these long protocols which need to be used in order to identify the silver strains. Well, yes, for us, we simplified a little bit the scheme. The sampling which need to be do it in the late spring. Then we are using the high RNA yield and higher purity by using this higher qPCR by using the robot, as I said before. Then we have a series of RT-PCR, real-time PCR, but we introduced the membranes. Instead of extracting directly the RNA from plant material, we included the membranes which we used and then processed by the same protocol for the moments. But we are trying to set up these props in order to use directly the props and to simplify this protocol. This is all. So I hope that I was not so fast.
Dr. Jalot, thank you so much again for another really interesting and insightful presentation. I just want to remind everyone that the chat box and Q&A section is open. So if you have a question, please do feel free to type a question for Dr. Jalot at any point. Dr. Jalot, just while we're waiting for questions to come in, I had one or two questions that came to mind just when I was listening to your excellent presentation. The first was connected to the kind of environmental factors that influence the severity of the strain of the virus. And I was just wondering, how do environmental factors such as like temperature or soil conditions, etc., influence the severity of the CTV strains and their detection?
First, I will thanks for your questions. First, I would like to say that there is no influence of temperature and like these irrigations or other environmental aspects. The unique environmental, let's see, between break that as an environmental as the insects. Insects, it was confirmed that insects they are transmitting, which during the transmissions they can have a role in having mutations of some genomes of CTV.
Excellent, Dr. Jalot. Thank you so much for that. Regarding techniques and tools, my other kind of question was, what would be the limitations, do you think, of current diagnostic methods when it comes to distinguishing between severe and mild strains of CTV, and how do you think they might be improved?
The current situation is, let's see, complicated because we are still using it. We are using several tests, and by eliminating, let's see, those who are not reacting, as I will show you now. You can see here, for examples, you have a series of tests which you need to adopt practically And because we don't have the others possibility to distinguish these silver strains, we have we have the tools, but the tool it's very long protocol to in order, and this protocol is used at research level. But when you are monitoring, for example, in the field, let's see, hundreds of samples, 200 of samples, you have an increasing of cost, increasing of time. So it's impossible to set up a monitoring in the fields with this protocol. This is why we are trying to do something in order to substitute these protocols with the simplest protocol, because we need it for the monitoring in the field. We need it in the area where these silver strains are not already introduced, or you have only, let's see, small area which these silver strains were introduced in order to uh, make a control of this disease. You need to test and then to practically uproot, to eliminate, to destroy the positive trees. And you need to have continuously this monitoring.
Very good. Thank you very much, Dr. Shalua. Just a couple of questions coming in from our audience. So the first reads, based on your experience, what is the best tissue that could be used for RNA extraction, and which RNA extraction do you recommend?
Uh, RNA extractions, as I said, this is what we are recommending. These are semi-automated magnetic paints. If it will not be possible because it's for the cost and for if some laboratory can't have it, we are still proposing the silica capture. Whereas for the membranes, you said, the second question, it was related to,
And
is
the
it
second
for RNA
question, so,
extraction?
and
Okay.
which RNA extraction do you recommend?
The RNA extractions are mostly mature leaves and from the vein of the leaves, using the
Excellent.
vein of the leaves. Yeah.
I see. Okay, excellent. And just another question here, just finally from another audience member. Dr. Dejula, great work and nice to see you. Have you considered using HTS instead of the PCR workflow, or using a LAMP-based test for a field test?
Yes, for the first HTS, no. It's increasing the cost also of the work, and it will be impossible, especially in this area for the Europe. Maybe it can be possible, but for some countries belonging to the South Mediterranean area, it will be complicated. And the second,
And the second was, it just was a follow-on, I think, or using a LAMP-based test for a field
yes,
test.
yes, the truth is that we already used this test. Yeah, the lamp, and we are trying to develop in parallel to test these different uh, let's see, sequences which which the Spanish partner will provide us with a lamp PCR. So you are going in this street.
In that direction. Excellent. And Dr. Gillot, thank you so much. I think we'll just leave it at that now. It's pushing up on 3 p.m. Irish time here.
Mhm.
I'd just like to thank you very much once again for an excellent presentation. And indeed, thank you very much to Dr. Wang for giving us wonderful and insightful presentations, and I think brilliant overviews of your respective projects this afternoon. Our next webinar will be on the 6th of March at the regular time of 10 a.m. Central European Time, when we'll continue our new webinar series on plant health and protection. I'll be sending the invitation out to all of you in the coming days. In that Breakfast Club, we will have Dr. Marika Samera, a senior research scientist at Tallinn University of Technology in Estonia, discussing the diagnosis and epidemiology of viruses infecting cereal crops, and Gudrun Strauss, a researcher at the Austrian Agency for Health and Food Safety, talking about the epidemiology and distribution of invasive arthropod pests in fruit crops and grapevines. Again, thank you very much to our two panelists today, and thank you everyone for attending the Breakfast Club webinar and for your questions. We really hope you enjoyed the webinar, and we hope to see you all again with us in 2 weeks' time. Thank you very much, everybody.
Thanks. It was a pleasure.
Thank you. It was a pleasure too. Thank you. Take care. Bye-bye.